Hi, everyone. So today we're going to talk about mRNA vaccines. mRNA vaccines have been in development for quite some time now, and they are considered to be the vaccine of the future. And so right now, if you've heard in the news, the latest information on COVID-19 is that they have a phase one trial that was very successful uh, regarding a vaccine against the coronavirus. And that particular vaccine happens to be an mRNA vaccine. And so we're going to talk a little bit about how these work and what this means for us because this is really like cutting edge science. This just came out last week. Okay, so first off, about vaccines. Okay, there are three different types of vaccines out there. There's the RNA vaccine, which we're going to talk about today, but there is also the antigen vaccine and the antibody vaccine. And those two, the antibody and antigen vaccines, they've been around for a while. Okay, and the idea behind these is that we want to trigger the immune response of our bodies. We want our bodies to have some mechanism for creating permanent immunity so that we don't get sick from all of these things and diseases that are out there. Okay, so an antibody vaccine is an interesting one because this one was developed, in particular with tetanus. This is an example. These were developed without making the body do anything. And so, really, what this is just a vaccine that has antibodies. We have found out that you can take antibodies from different individuals and transfer them to other individuals and help those people be immune to things. And so, this is actually what's been going on with people who test positive for the antibodies for coronavirus. That's one of the biggest things they're looking at right now is how many people actually have antibodies because that means that they were exposed to it and they were able to successfully fight it off. So what they're doing with those antibodies is they're asking people to donate their blood plasma so that they can extract the antibodies and give those to people who are seriously ill with COVID-19 in the hospitals. And the idea is that by presenting people with these antibodies, they can help shorten the length and the severity of the infection. So by giving people these antibodies, they're able to recover faster and they generally have fewer complications. And they're also more likely to be discharged from the hospital and get home and recover without any complications. An antigen vaccine is a more traditional vaccine where you get a weakened form of the virus. They heat treat it, they kill it with chemicals. And so what you're left with is this outer protein coat of the virus or specific proteins from the virus. Okay. And that triggers our bodies to produce an immune response. And then the third one here is this RNA vaccine. This contains messenger RNA, right? Messenger RNA codes for viral proteins. So what they're basically doing is they're taking the genes from the virus that code for a specific protein and injecting that into our bodies. Okay, so let's take a look at antibodies real quick in terms of the vaccine. So an antibody vaccine is produced by another individual. Okay, we know that. And the antibodies are extracted from the blood plasma, and then we inject those antibodies into the bloodstream. Okay, and that allows those antibodies, once they're in the bloodstream, to attack any type of antigen that we may encounter. Remember, antibodies attack antigens. Antigens are the things that might cause disease. Okay, an antigen vaccine, just like an antibody vaccine, is injected into the bloodstream. Okay, and the same thing happens with RNA, right? We inject that, and the RNA enters the cells, and it produces viral proteins. An antigen vaccine gets injected into the bloodstream. It's going to trigger the production of antibodies. Okay. Now, one of the things with an antibody vaccine is the immunity is temporary. So when you get a tetanus shot, that tetanus shot only lasts about 7 to 10 years, and then we have to get another booster of antibodies. People that receive the COVID-19 antibodies from other people that are in the hospital, um, those antibodies don't last forever in their system. It gives them immunity. They're not sure for how long because we're only a few months into this thing, but it does provide them with some protection and some immunity when they come home from the hospital. Okay. An antigen vaccine has always been the best way to ensure that people get long term immunity because this triggers our body to produce antibodies against a virus or viral proteins. That means we have our own immunity to it. It's called innate immunity and that immunity is long lasting and often it's permanent. Now, sometimes you need boosters of antigen vaccines. Like when we were kids, we all had to get our measles, mumps, rubella vaccines. And that helps to ensure that the body mounts a significant immune response so that we have that long-lasting permanent immunity. If you miss a vaccine, you may not be completely immune, and that immunity may wear off sooner rather than later. Okay. And then with the RNA vaccine, once that RNA enters the cells, it produces viral proteins. Those proteins are released from the cells and they enter the bloodstream. So now we have these little proteins from the virus that are in our blood. And what that does is it triggers antibodies to form against the viral proteins. This gives us permanent, long lasting immunity. Okay? And the nice thing about an RNA vaccine is you don't have to worry about introducing the whole virus or parts of the virus into the bloodstream. This goes right into the cell, and the cell immediately starts producing this. So our bodies will always produce this protein and will always produce antibodies against this protein. So that gives us much better lifelong immunity. Okay. Now, the way the coronavirus works is it has these spike proteins on the outside. Those are the antigens that help the virus spread. And the virus spreads specifically by using these spikes to bind to these receptors. They call the ACE2 receptors on the cell surface, on the plasma membrane surface. And what the cell does is when the virus attaches to these receptors, it's like a key opening a lock, and it triggers the cell to pull the virus in. So the cell basically welcomes the virus in once it attaches to these receptors. So we can neutralize these spikes by blocking them using antibodies. So the antibodies that are produced in our own cells, if we were naturally infected with this and had to deal with all the symptoms and the severity, we would produce antibodies to eventually fight off the virus and destroy it. And then that would give us lifelong immunity. But people don't want to get sick. And so the idea is to develop a vaccine so that we can make these antibodies on our own without all the issues of illness. Now, the antibodies are specifically produced by white blood cells called B lymphocytes. And the antibody you can see here, it's a little Y shaped protein molecule that very specifically targets these spike proteins, and it'll completely neutralize the virus. Okay. So an mRNA vaccine works by introducing an mRNA sequence into a cell, right? And this tells the cell what to build. Specifically, in this case, it's going to tell the cell to build the COVID-19 spike protein, okay? Once it builds those spike proteins, and the cell codes for those proteins trigger antibodies to form, which recognize the antigenic proteins on the surface of the virus. Okay, and then the cells of our body, the idea with this is that the cells of our body will continually make these antibodies, and that confers long-term resistance to these infections so that you're permanently immune. Okay. So the question is how to get the mRNA into the cell. So the problem with mRNA is it's unstable and it breaks down quickly. So it has to be delivered into the cell in such a way that it can survive the trip. Okay, not only from the syringe into the cell, but then once it's inside the cell, it can do what it needs to do. And it also has to be able to get through the cell membrane. So we know that the plasma membrane or the cell membrane is made up of phospholipids. So what scientists have done is they've developed these lipid nanoparticles, and they've been utilizing these as a way to deliver things into cells. Okay, and it's known as encapsulation, right? So the messenger RNA gets encapsulated into this lipid nanoparticle, and that lipid nanoparticle particle has the ability to enter the cell because the cell membrane is made of lipids, and lipids can fuse with lipids very easily. Okay, and so that lets it enter the target cells. Once inside the cell, this lipid nanoparticle quickly disintegrates, and then the mRNA gets translated into active proteins, specifically the spike protein. Okay, and so the picture here shows you this is a cross section of what that nanoparticle would look like. But if you looked at it from the surface, it would just be this ball of lipids that allows it to enter cells. Okay. And so once it enters the cell, it gets pulled into the cell through what's called an endosome. Endo means into, and zome means opening. All right. And so it pulls it in, and then this particle that has the spike protein messenger RNA in it disintegrates. Ribosomes read the messenger RNA and they produce a protein. And that particular protein is the spike protein, which gets released out of the cell into the bloodstream. And what happens then is white blood cells, the B lymphocytes, identify these as foreign proteins and they start making antibodies against that protein. And within a few weeks, we have a completely well armed immune system that can now readily produce antibodies against COVID-19. Okay. So there are three types of RNA vaccines. One of them is called non-replicating mRNA, and this one is the simplest form of RNA, where you just package it and deliver it into the body, and the cells make the antigen. There's another one called in vivo replicating mRNA. This is where you package it with additional RNA strands, so ensure that it's going to get copied once the vaccine is inside the cell. This also is more efficient because this means that you're going to get greater quantities of antigen made from a smaller amount of vaccine. Okay, and this means that you're going to get a lot of protein being produced from a small amount of vaccine, which triggers a stronger, robust immune response. This is what the current mRNA COVID-19 vaccine is. And then there's another one here known as a dendritic cell non-replicating mRNA vaccine. And this one is where they take dendritic cells in the lab and they expose them to the RNA. Okay, and so what they do is they take these immune cells that present antigens on the cell surface. They extract them from the patient's blood. They put the RNA vaccine in them, and then they give it back to the patient. So it's a little more complicated. It's a little more tricky, which means it costs a little more money and it takes a little bit more time. So if we're looking at the most efficient way to do this, RNA vaccine number 2, this one, the in vivo self-replicating one, is the best one. In vivo means in the living organism, within the organism. In vitro means in the lab. So this would be a little more time consuming because you have to get these dendritic cells, present them with the antigen, extract them from the blood, um, put the RNA in them, and then put them back into the patient. Okay, so there's a lot of back and forth here and a lot of things that could potentially go wrong. So number 2 seems to be the best route. Okay, what is the benefit of an mRNA vaccine? They're safe because they're not made with the pathogen particles or inactivated pathogen. So they are not infectious at all. And the RNA does not integrate itself into the host genome, right? The RNA strand in the vaccine gets degraded once you make the protein. So once we produce that protein and the protein gets released into our bloodstream, you don't need that RNA anymore and it gets broken down. Efficacy means how effective is it? Early clinical trials are showing that these generate a really strong immune response, and there are very few, if any, side effects, and you can produce them much more rapidly. And that's the idea. We want to produce a vaccine very quickly, especially in this situation, so that we can get it out there and respond to this outbreak. Okay. Okay. And so here's some information. They gave participants 2 doses um, of this vaccine from 25 micrograms to 100 micrograms. And what this showed is that all of them seroconverted with binding antibody levels at or above levels seen in convalescent sera. So, what does that mean? That means that after 2 doses, these people produced antibody levels that were equal to or above the antibody levels seen in people who were actually infected with the virus and got sick. Okay. Um, what they've found out with this particular vaccine is that it produced neutralizing antibody titer levels. Antibody titer means concentration of antibody. So this means that it produced a level of antibody that was enough to neutralize an infection in all 8 initial participants. Okay, and that these people reached or exceeded neutralizing antibody titers generally seen in people who are infected. That's a great thing. Um, it's safe and well tolerated. And what they did with this is they took this vaccine, they gave it to a mouse, and then they exposed the mouse to COVID-19. And it provided full protection against viral replication in the lungs of a mouse challenge model. So that's a really good sign. So after they injected this into a mouse and let the mouse build up its antibodies, they then exposed the mouse to the actual virus, and the mouse did fine. The mouse was able to be completely protected. And so the next phase, this is phase 1. They're looking, and they did phase 2. They're looking to do a phase 3 study in July with many more people. Okay, and the hope is that they'll be able to really get this thing moving along and show some positive results. So this is a really hopeful situation with this vaccine. I'm just going to go back for a second. The vaccine itself is called mRNA 1273. So if you Google that and look it up, you'll find up the latest information about it. But it's really promising that this can be developed quickly and effectively and safely, and that it triggers an immune response between those ranges. And again, 25 to 100 micrograms is real tiny. Okay, you don't need to use a lot of this RNA in the vaccine in order to trigger an immune response. What they found out is they did this at higher levels, and it actually triggered such a strong immune response that people started to develop symptoms as if they were sick, but they really weren't. But they started to feel miserable. They got the cough, they got the congestion, they got fever. And so they realized that they can use a smaller dose and still get the same amount of antibody response um, with the lower dose. And so it's very promising. So if you watch the news and keep up with this, you'll see some additional information. And over the summer, we should be hearing some more information about the success of this. If this works well, this very well could be the vaccine that is being utilized to help control and stop the spread of the infection of the coronavirus.